So to give you guys a bit of an overview, today I'm going to take you guys through genetics, which includes chromosome disorders, population genetics, and SNPs. I'm going to try to get through that quickly because Ben, I think, is going to take you guys through even more biochem. Today it will be lipid metabolism, alcohol metabolism, and the great metabolic race, which I know you guys have been struggling with a little bit. So hopefully this will help you guys understand that a bit better. Okay, here we go. I'm going to share screen now. I can get that to load. Is that working for everyone?
Just put a thumbs up if you want.
Yeah, that works.
Cool. Okay. All right, so firstly, we're going to start with population genetics. Some definitions to start with. So a trait is a characteristic that humans can have. And here I've put eye color and hair color, which are physical traits, but you can also have things that are biochemical or physiological, such as, you know, a certain protein, or whether you produce an enzyme, or when in your life you produce that protein or enzyme. So a phenotype is the manifestation of that trait. So how your body manifests that particular trait, basically. So for eye color, it might be brown eyes or blue eyes. For a certain protein, it will be whether you produce that protein at all, or do you produce it in early life or towards, you know, middle age or whatever. A gene is a region of DNA that influences a trait. Simple enough. An allele is alternative versions of the same gene. So the DNA sequence will be slightly different for each allele, whether that is different by one base pair or a longer segment of DNA. So your genotype is which combinations of alleles do you have. So as you guys probably know, you can only have 2 copies of each gene. So for every gene, you can only carry 2 alleles. And the combination of the alleles that you can have is your genotype. So for a gene that only has 2 alleles, it will be, here I've put big A and little a. So if your genotype is both little a or both big A, then you're considered a homozygote. And if you have 1 big A and 1 little a in your genotype, then you're considered a heterozygote. And a locus is just where on the chromosome the gene is located. So when we're talking about population genetics, we're considering the frequency at a population level rather than just looking at one individual. So phenotype frequency is the number of people in the population that has a certain phenotype. So this is the equation here, simple enough. Similarly, genotype is the proportion of people that have a certain genotype. So number of people with genotype over the total number in the population. Allele frequency looks a little bit more intimidating, but it's okay, we'll break it down. So the frequency of allele is represented by P or Q, and this is considering a gene that only has 2 alleles. So yeah, like I said, for this, we're going to consider a gene that only has 2 alleles, and allele big A is dominant, and allele little a is recessive. Whoop. I'm literally turning into Craig acid. All right, so for phenotype and genotype frequency, this is pretty simple. So I'm going to challenge you guys a little bit. So what frequency am I considering with this data here? Drop your answers in the chat. Try and see. Bring up the chat. So drop your answers in the chat. Which frequency am I considering, A, B, or C?
Did you specify which one you're looking for?
Okay, so we're given this data here. The numbers with the specified genotype. So which frequency in this population of 100 individuals would we be looking at with this with this data? Yep, so some people are saying B, which is correct, genotype frequency. Okay, so if I All right, so we're going to look at phenotype frequency. For this one, we're going to look at people with brown eyes. So if I tell you that the allele big A codes for brown eyes and is dominant, and the allele little a is coding for blue eyes with and is recessive, then which genotypes would I look at to find the people with brown eyes? Why doesn't this work? There we go. So gotcha. Yep, you guys are saying big A, and big A, little a, which is perfect. Right. There we go. So this is just calculating the frequency of the people with phenotype brown eyes. So, perfect. This looks very intimidating. Let me break it down. Okay, so allele frequency is the thing that you're going to be, that looks kind of intimidating, right? So here is the equation. Okay, here we go.
Screaming ensues.
I know. Okay, so here is the equation. Now, the best way to think about the allele frequency equation is to think of it as how many copies of the allele is present within the population. So if P represents the amount or how many copies there are in the population, and big NAA here is representing the number of people with the genotype big A, then for each of these people, they're carrying 2 copies of the allele big A. And so for every person, there's going to be 2 copies of the allele big A. And that's why you double that number. Whereas for people with the genotype big A, little a, they're only going to carry 1 copy of the big A allele. And so you don't need to double that number, because for every person, there's only going to be 1 big A allele. So the 2n here, n is standing for the total number of people in the population, and you're going to double that number, because for every person there will be 2 alleles. So the total number of alleles within the population is going to be twice the number of individuals in the population. So if you plug in these numbers up here into the equation, you're going to find out that the amount of the population that carries the big A allele is going to be 46%. So in the participants section, can you thumbs up if that makes sense? And if you want me to explain it again, but slower, then hit the go slower bit. Maths does suck, but it is doable. Does that sound okay to everybody?
Yep, seems like people are getting it.
Yeah, cool, perfect. All right, so a note on allele frequencies, the amount of, so every gene will have a different amount of alleles. Usually it's 2, but it can be more, but the frequencies of all the alleles should add up to 1, right? And if you just think about it mathematically, you can't get a proportion, like, it's just the proportion of people who have certain alleles, and you can't get to more than 1. So mathematically, it makes sense. All right, so these are just some questions. I don't know if we're going to have time to go through them today, but feel free to have a look at them after the session. And if we have time at the end of the session, we might go back to these. All right, the Hardy-Weinberg equation. So what is it? The equation is 1 is equal to p squared over plus 2pq plus q squared. So p is representing an allele frequency of A, q is of big A, sorry, and q is representing the allele frequency of little a. So this is again working off of the assumption that this gene only has 2 alleles. So the Hardy-Weinberg equation says that in a population, the allele frequencies of this allele will not change over time if the population is randomly mating and free from the factors of migration, mutation, selection. So why would these change it? Basically, these are going to change the allele frequencies in a couple of different ways. Mutation allows for the development of new alleles. So if you introduce a new allele, then that's going to change the frequency or the proportion of people in the population that has the previous alleles, right? Migration will allow movement of alleles into and out of the population. So again, that's going to change how many people have the allele within the population. Selection, so the natural environment is going to allow some alleles to live or survive better in that environment, or it's going to select against that allele. So that allele cannot survive as well in that environment. And non-random mating. So non-random mating would be selecting specific individuals with a specific phenotype or genotype to mate with. And that will result in an increase in homozygosity or increase in heterozygosity. So it changes the genotype frequencies and as such the allele frequency as well. Random mating would be just looking at a population and going, I don't know, that one, I guess, and choosing completely at random rather than choosing for a specific phenotype or genotype. All right, so how is the Hardy-Weinberg equation relevant? You can use it for some different things. So if you are given allele frequencies, you can predict the genotype frequencies for that population. And if you have the expected genotype frequencies for that population, you can compare it to the actual observed genotype frequency within that population and see if that matches up. So if the observed genotype frequency matches the expected ones that you've calculated, then that population is not being affected by these factors of migration, mutation, selection. But if they don't match up, then obviously there is a factor at work that is affecting the allele frequency or the genotype frequencies. You can also use the equation to estimate the carrier frequency, which is related to calculating the genotype frequency. Carrier frequencies is basically when you are heterozygote and you carry one copy of a disease-causing allele that is recessive. So you don't have the disease, but you might pass it on to your offspring who can then inherit the disease. And so you can estimate how many people in the population is actually a carrier for this disease. So again, this is just some equations that are illustrating the usage of the Hardy-Weinberg equation. So you can predict genotype frequencies if you are given allele frequency. You can be given some genotype frequency and assess whether the observed genotype frequencies that you are given match the genotype frequency that you can calculate using the Hardy-Weinberg equation. How would you do that? Well, firstly, you would have to calculate what the expected genotype frequency is, right? But to do that, you need to calculate the allele frequency. So you calculate the allele frequency using the allele frequency equation that we went through earlier. From there, you would use those allele frequencies to calculate expected genotype frequencies using the Hardy-Weinberg equilibrium equation. And with those genotype frequencies that you've calculated and are expecting, you compare that to the actual observed genotype frequencies and see whether they match or not. And then you can use that to speculate as to whether there are factors that are affecting the population and what those factors might be. Does that sound okay to everyone so far?
Just to cut in for a second, like some of you will be actually
Sure.
wondering, why is this actually, like, you may be confused because you think, in nowhere in the world does this actually fit where nothing actually affects the population. And yes, you're correct. So the point was that we can also use this equation to see if there is no progress being made as well. So the point is more not to use as in to see there's nothing happening in the world in terms of mutation or allele frequency change, but it's more to say that if we look at these diseases, how are these changing, and where might the factors actually be that are affecting these changes?
Yeah. So yeah, as Brendan said, it is very rare that a real life population will have no factors affecting it. So yeah. So the third thing you'll notice that we mentioned that the Hardy-Weinberg can be used for is estimating carrier frequency, and this is going to come in much more relevant when you think about genetic counseling. So if you have a mother who's pregnant who wants to know if her child is going to have a disease, then we can use the disease incidence that we've observed from epidemiology and the inheritance pattern of the disease and work out how many people in a population is likely to be a carrier. And that will give you a indication of whether the mother will be a carrier or not. This will obviously be different if the mother has a family history of the disease. This will increase or decrease her chance of being a carrier. So speaking of inheritance patterns, again, this is assuming a gene will only have 2 alleles. So when you're looking at a pedigree, which you might be asked to do in the exam, when you're looking at a pedigree to determine whether or what the inheritance pattern is, there are going to be 2 factors that affect the inheritance patterns. So whether it is transmitted on an autosomal chromosome or a sex chromosome, and whether it is a dominant allele or a recessive allele. So autosomal conditions or alleles on autosomal chromosomes will affect male and females equally, whereas sex chromosome linked alleles will affect one or the other more than the other. So it will either affect females more or males more. Dominant alleles will never skip a generation because only one copy of that allele will give you the disease, whereas recessive alleles can skip a generation because you need both alleles, and there's a chance you can be a carrier instead of having the actual disease. X-linked dominant alleles will affect females more because you only need one copy of the allele to be affected, and females have 2 X chromosomes, and so they have twice the chance of having that allele, basically. And affected fathers will always produce affected daughters because if the father is affected, his 1 X chromosome has the diseased allele, and he will definitely pass it on to his daughter. And because you only need 1 copy of the affected allele to have the disease, that means that his daughter is basically guaranteed to have the disease, unfortunately. X-linked recessive is kind of the opposite, actually very much the opposite. Males are more commonly affected because you need both your X chromosomes to have the disease carrying allele to be affected. And in males, males only have 1 copy of the X chromosome, and so only 1 copy is enough to give the disease to the individual. So affected mothers will always produce affected sons, because if you are a mother and you are affected, that means that both of your alleles are the disease carrying allele, and you will definitely pass one of those to your son. And because males only need 1 copy of the allele to be diseased, then that means that the male is guaranteed to have the disease. Y-linked will only affect males because only males have the Y chromosome. And if your father is affected, that means that he has the disease carrying allele on his Y chromosome, and he will pass that chromosome onto his son. So his son is guaranteed to have the disease. All right, so I hope that makes sense. We're gonna go to SNPs real quick because there's not much in it. So what is a SNP? A SNP is not a mutation. A SNP is only a differing point in a genome. So at a certain point in the genome, some people might have an A, some might have a T, some might have a G, some might have a C. But it is not necessarily causing a disease. It's just different between different people. It's only considered a SNP if more than 1% of the population has it. So you can have 1.5% of the population have a G at that point in their genome, and it is considered a SNP. But if 0.5% of people have it, or if 0.9% people have it, then it's not a SNP. And this can occur anywhere in the genome, within exons, within introns, or within DNA segments that are in between genes. So it can occur anywhere. So SNPs don't give you a disease. So that differing base pair does not guarantee you a disease, but it increases your risk of having a disease. So one of the examples of that is on chromosome 19, there is a particular SNP, and a certain combination or a certain base pair will give you an associated risk of Alzheimer's disease. But this will only increase your susceptibility. It will not definitely give you the disease. So if you can correlate the presence of a SNP with the presence of a certain disease, you can start finding a correlation between those two things. So how would you do that? With a genome-wide association study. So basically you take a lot of people with that disease and a lot of people without that disease. You examine all of their genomes and see which SNPs are in their genome, and then compare which SNPs are in the affected people versus the not affected people. And from there, you can start to correlate whether a SNP is correlated with a certain disease or not. So that's basically all that you really need to know on SNPs. There is some things about medication, about how SNPs influence your body's interaction with the medication, but that's an aside, and you don't really need to focus on that too much. All right, does that sound okay to everybody before we move on to chromosomal disorders? Cool. All right, so chromosomal disorders. We're going to set the scene a little bit by talking about chromosomes. So the structure of a chromosome. So it starts here with DNA strands, which are going to wrap around protein balls that are called histones. And this structure here is called a nucleosome. So nucleosome is going to start forming the structure of like beads on a string. And these beads are going to form little coils here, which forms into denser coils, which forms into even denser coils, which eventually form into a chromosome. Yep. So with chromosomes, you have, so this is an unreplicated chromosome, and this is a replicated chromosome. You've got the centromere in the middle, the P for petite arm, which is the shorter arm, and the long arm, which is called the Q arm because Q comes after P. And the telomere is the tip of each arm. They can be classified according to where the centromere is. So if it's in the middle, it's called a metacentric chromosome. If it's kind of slightly towards one end, it's called a submetacentric chromosome. And if it's sort of all the way towards one end, it's called an acrocentric chromosome. And this bit on top here would be called a satellite. So you can see that your own chromosomes are kind of organized based on whether they are submetacentric, acrocentric, or metacentric, which is just a fun thing that I thought was kind of neat. All right, so how do you study chromosomes? Basically, you take a cell that is in the middle of dividing, and at prophase, which is when the chromosome is at their most dense, you inject a chemical called colchicine, which is going to prevent the cell from dividing further. So you stop the cell at the moment when the chromosomes are at the most dense and most visible, so that you can see where the chromosomes are. And to organize them further, you use G-banding or Giemsa banding, which is basically a type of stain. It's going to produce some lines, some bands here. The light bands are where the genes are most active, and the dark bands are where the genes are least active. So each chromosome is going to have a very characteristic set of bands. And you can use that to organize this mess here into paired chromosomes based on their length and on their band staining. So a karyotype is a full set of chromosomes in an organism. It usually looks like this in a little diagram. But it also can refer to this kind of notation here. Now, the notation is ordered in a very specific way. You start with the total number of chromosomes. So here you can see 46. The sex chromosome makeup, which tells you the gender or the sex of the person, and any observed abnormality. So here you can see 46 XY, which is normal male. 46 XX is a normal female. 47, you can see there is an abnormality. There's an extra chromosome. XX means female. And here you see the observed abnormality is plus 21. So they have trisomy 21 or Down syndrome. So we're going to, oh no, I want to go back. There we go. So the nomenclature, the most important ones are P for short arm, Q for long arm, deletion, insertion, and inversion. Those are the most important ones that you want to keep in mind. So with that, chromosomal abnormalities. All right, so there are two broad categories, numerical and structural. So within numerical, you've got aneuploidy first, which is a missing or additional set of one or sometimes two chromosomes. So you can either have monosomy, which is one less chromosome, or trisomy, which is one extra chromosome. Rarely you will have tetrasomy, which is extra two chromosomes. So there's only kind of 5 aneuploidies that make it to birth, and 3 of them are autosomal aneuploidies. So you've got trisomy 21, 18, and 13, Down syndrome, Edwards syndrome, and Patel syndrome, respectively. And the way I remember this is kind of backwards, but if you think of Down, Edwards, and Patel, that's going in alphabetical order, and it kind of goes backwards so that the one that comes first in the alphabet is the name of the largest number. So 21, 18, 13 goes down the alphabet, I guess. Trisomy 18 and 13 will make it to birth, but the child will not make it past the first year. And even within the first year, there are profound developmental delays. In contrast, Down syndrome will be able to make it past the first year, even with the developmental delays and the impairments and the other symptoms associated with the syndrome. And this is because 21 is one of the smallest chromosomes and it codes for the least proteins. And so there is, to an extent, you can survive without those proteins. All of these are affected by maternal age. So the older the mother at the time of conception, the more likely the child will have one of these syndromes. So you can see the karyotypes here, 21, 18, and 13. So the sex chromosome aneuploidies, Turner syndrome is a missing sex chromosome, and Klinefelter's is an extra X chromosome. So Turner syndrome will occur in female births, and I kind of remember this by remembering Sophie Turner, who's a female. So yeah, that's how I remember it. And Klinefelter's will occur in males. And each of these have their own symptoms and their own treatments, so you can have a look at that later. Yep, so karyotypes, that's Klinefelter's, that's Turner's. So the other type of numerical chromosomal abnormality that you can have is polyploidy. So this is not just an extra chromosome, but an extra set of chromosomes. So instead of having 2 copies of each chromosome, you have 3, sometimes 4, but probably not. Unfortunately, none of these are viable. They won't even make it to birth. The embryo will likely abort spontaneously. There's this one in the chat.
Do we need to memorize the treatments for the disorders from acupuncture? No, I don't think so. It's more just to say that you know what it is. At this point, we're not really treating anything genetically. Yeah.
I think the most likely thing you're going to get is like they'll give you a karyotype and ask you what disorder or what chromosomal abnormality it is.
Yeah, so they'll give you like the image and you just have to see.
Yeah. They might also ask you to do the labeling of it, so like the 46XX, and then
So you have to pick the right one.
Yeah. Cool. So structural abnormalities. These will result from the chromosome breaking and then rejoining at a abnormal place, so a place they shouldn't be joining in, and most commonly in meiosis at the crossing over and recombination stage. So there are several types. Translocation is when a section of chromosome on a section of DNA on one chromosome will move to another chromosome. A reciprocal translocation is when two chromosomes swap an equal amount of DNA. And this means that the rearrangement is balanced, so there's no net loss or net gain of DNA material. So it will usually be harmless in the individual, but their offspring will have a partial aneuploidy. So aneuploidy of just that DNA section where there's an extra bit of DNA or a loss of that bit of DNA, but it won't be an entire chromosome, if that makes sense. So their children will be affected, but not the individual themselves. The other type of translocation is Robertsonian translocation. So this occurs in two acrocentric chromosomes, so that the longer arms here are going to fuse at the centromere, and then the satellites are going to be lost. So this most often occurs in chromosome 14 and 21, and it will lead to trisomy 21 in the affected individual. There is something called an insertional translocation, where a section of the chromosome is inserted into another chromosome, and there's no swapping. So this is kind of an insertion as well. So you can consider it as an insertion rather than a translocation where it's a swap. The other abnormalities are deletions. So one bit of the chromosome is deleted. Duplication, so a bit of the chromosome is replicated twice. And an inversion, which is better seen in this one actually. So the section of DNA is replicated upside down. And these will have their effect if the breakage is in the middle of a gene that codes for a particular protein, which will have a physiological effect. Or if this section that's been deleted contains the gene for an entire chromosome, the gene for an entire protein, so that protein won't be produced by that chromosome. It needs to be the other copy of the chromosome that's producing the protein.
A question from Karina.
So, yeah.
Yeah, I just want to ask for the Robertsonian translocations. It's not what's happening actually.
Sorry, what was that again?
Oops. I mean, for the Robertsonian translocation, is it that they kind of like break up from the centromere and then join together?
Yeah, basically. So you see the dotted line here, that's basically the chromosome breaking at the centromere. And instead of joining back to the right, what is the word? Satellite here, it joins to the other chromosome instead. So you've got, if this is chromosome 14 and this is chromosome 21, you've got chromosome 14 joining to 21 and forming a metacentric chromosome here. And then these two bits just get lost.
Oh, okay, cool.
Does that make sense?
Yeah, that makes sense. Thank you.
You may need to know the name of those like little two bits after they also get lost as well. So that I think they call it satellite. Was it satellite? Satellite pieces or something.
Yeah.
They may ask you like they may give you one multi-choice on that as well. So that's something you may need to know. I think it's satellite. Just like bits of lost satellite DNA or something. I'll get back to you on that.
Satellite sounds right.
Okay, thank you.
All right. So I think that's it for types of rearrangement. So you can have balanced rearrangement, which is when there's no net loss or gain of chromosomal material. So this is harmless, but the offspring of the affected individual will have an aneuploidy for just that bit of chromosome. Whereas unbalanced rearrangement is when there is net loss or gain of the DNA segment, and so they usually have very severe clinical effects. I'm going to go back to this quite far back. Nope, even farther back. This slide here, you can see the bottom here. If you look at the karyotype, 46, which is normal, XX, which is normal, but you've got the observed abnormality, which is deletion in the P arm of chromosome 5. So I just wanted to let you guys get used to the notation here. All right, and I think that's it for me. Does anyone have any questions before we move on to Ben's part of the presentation?
I have one. Can you make me co-host?
I
Why?
made you co-host before you left, Sabkan. So maybe you shouldn't have left.
Wow.
All right.
Okay.
Also,
Karina, do you have a
why?
question as well? Or
Um,
did you want to go back to the thing that you asked about before?
just now I have a question for like the 2p, like something showing like 5p just now. I can't remember
Yeah.
which slide is it. Like go back for a bit.
Yeah, I'm trying to go back.
Yeah, back.
So not that one, sorry. It's here.
Yeah. So what was that deletion 5p there?
Yeah, so this notation is to show you that del stands for deletion, 5 is for chromosome 5, and P is for the short arm of the chromosome 5.
Oh, I
So,
see.
So
like
you can
here,
remember P for petite.
yeah, and Q comes after P.
Cool. And then the other question I have is that about the population stuff, like p squared
Sure.
plus 2pq.
Yeah.
Yep,
The,
so I can move back to the Hardy-Weinberg equation.
Yep, that's the one.
Here we are. Yep.
Yeah. Do I have to remember this or?
I can't quite remember if it showed up in the exam.
It was in the exam.
It was in the exam,
You
1 or 2 questions.
Questions.
Yep.
There's 1 or 2 questions on it. It's really like easy, so I recommend just learning it so you get those questions right.
Yep.
Flex harder, I dare you.
And this is basically just the quadratic formula, but instead of 0, it's 1. So, yeah, the formula itself is easy to remember.
That's
You
not a
just got to
quadratic formula.
yeah.
Right,
So
go on,
you
go
just
ahead.
need to remember what each of these stand for. Yeah, and from there, you will be able to, you know, calculate what the. I reckon this is basically all you need, right? Like.
Was it, sorry, Ben, was it like binomial expansion or something, right?
What?
Don't worry.
Math sucks.
Expanding this.
Just keep going.
Yeah.
I would also recommend going over the examples in the lecture and like trying to work them out yourself, because that's what I did and that helped me exam time. So I do recommend learning it.
Okay, sure. And I just want to say that like p, like with this stuff there, is it time 2 or to the power of 2?
To the power of 2. I couldn't figure out how to make it like upper script. So yeah, it's to the power of 2. Not
But isn't that when we were calculating genotype frequency before, we were like timesing 2, or is it a different stuff?
So that was for allele frequency. So if I go down to this one, so you can see that the allele frequency equation you can use to calculate allele frequency when you're given the genotype frequency. Whereas the Hardy-Weinberg equation is the opposite. So you use it to calculate genotype frequency when you're given an allele frequency.
Oh, I see. Is it because they can like arrange differently, so we have to like to the power of 2?
I think it's just to do with like what information you're given. But if you're given allele frequency, you know that you need
You calculate the allele frequency, right? They'll probably give it to you, but for example, if they do it from scratch, they'll calculate the allele frequency, so they'll use the bottom left one, and then they'll use that and put it into P and Q, and then they will square that and put it into the equation to see whether there's been any changes like that have been made.
Okay, cool. And what does the 1 symbolize in Hardy-Weinberg equation?
So because these are the different genotype frequencies, right, within a population. So each of these will come out to be a fraction of the population. Because it's like what percentage of the population has this particular genotype. And so if you think about it, the entire population the entire number of people in the population can't go past 1. So all of the genotype frequencies should add up to 1. Does that make sense?
Yeah. If it's not 1, it means it's changed, or it's less or more. There's more there's more alleles. There's something else being introduced.
Yeah, and it's the same for allele frequency as well. So if allele frequency is the proportion of people with a particular allele, then all of the allele frequencies for a gene should add up to 1.
Cool. Thank you.
Right. Does anyone else have any questions? If not, then I might. Ben, do you have your slides up, or do you want to?
Yeah, chuck me screen share.
Okay, so I'll stop share, and then Ben will start his screen share.
Where is it?
Cool.
Ben, turn on your camera.
My webcam doesn't work.
What camera?
She doesn't work.
Big sound.
Is that working?
Yeah.
Yeah, that's fine.
Okay.
Up to you.
Just before we start, Khan and I have uploaded HK's pre-reading slides. Just gonna plug those. If you guys haven't seen, we just don't want you guys to suffer through the same pain. Khan and I.
Yeah. So HKS is tomorrow, so
actually do all the pre-reading. Please don't do all the pre-reading like we did.
Yeah.
Tell all your friends about
deserve.
it. Yeah. We just don't want Jappies to go through that same pain. Anyway, we're going to do a bit of biochem this week. It's a continuation of lipid metabolism because it was a bit rushed last week. Also some alcohol metabolism. I know you guys haven't done it yet, but you'll probably have a tune tomorrow, I think. So just a bit of like pre-learning, and you can go back to this whenever you want after YouTube. Just a bit of a recap from last week. This is just some good terms to know. One thing that's important, I guess, is like if you look at like the suffixes, so lysis, genesis, I think they'd be pretty helpful in helping you to memorize what those terms mean. So if you look at it, it says like glycolysis, right? So lysis basically means breakdown. So if we have lysis of glyco, it's like glucose, it means that you're breaking down glucose to something else, pyruvate. Whereas here, if you have genesis, right, that means like creating, right? So you have gluconeogenesis, that means that creating new glucose. So you're creating new glucose from pyruvate. Like same with here, so you've got glycogenesis, so you're creating glycogen or glucose. That's just a bit of a refresher and good way to help you remember. How do I? Okay, yep. Yeah, just a bit of a refresher on the terms in aerobic respiration. So that's basically how you get ATP from a glucose. So you go to glucose from glucose to pyruvate, and then pyruvate to acetyl-CoA, acetyl-CoA to NADH2, and that goes through the electron transport chain.
We've got a question.
I have a question. So I'm a bit confused about what the glycol mean. Does it mean refer to the glycogen or the glucose for the part glycol?
Glyco. Yeah. So.
It can be pretty confusing because I think you just have to memorize like which ones are which exactly as well. So looking at the endings will be more helpful than looking at the beginnings first of all. But glycogenolysis is the only one that actually means glycogen to glucose.
That one's pretty confusing.
I think glyco should be glycogen and gluco is glucose. Because if you look about glycogenolysis, that's breaking down glycogen. And glycogenesis, so glycogenesis is making glycogen. So the only one that's different is glycolysis.
So that's what got me confused. Is there a reaction that can convert like glucose to glycogen?
Yeah, so I think that's glycogenesis. I think that's just the one exception.
Yeah, glycolysis is like the only one that's different.
Yeah, like if.
And that could be due to the fact that it's a part of the cellular respiration steps. rather than maintaining blood
If
sugar.
they named it properly, it should be glucolysis.
But there isn't
Name
one.
it
Yeah.
properly, but they didn't name it properly.
It's a
So,
trap.
So
blame
yeah,
the scientists there.
glycogenesis. Yeah, birthing glycogen or making glycogen. So the bottom two should be correct. Just remember that glycolysis is metabolizing glucose.
And pyruvate is kind of like the intermediate part between glycogen and glucose.
So if we go here, right? So I think before this we should have like glucose, right? So before this we should have what's the word where you convert glycogen to glucose?
Glycogen to glucose. Glycogenolysis.
Oh yeah, glycogenolysis. Before this we have a glycogenolysis, right? So that's basically just okay. How did you do that? Anyway,
Nice.
yeah. So that's basically the process of converting glycogen to glucose, right? So if you see here, pyruvate would actually be the intermediate step between glucose and acetyl-CoA, right? So you have to go from glucose to pyruvate, right? And then the pyruvate you get will be turned to acetyl-CoA. Right? So basically it goes glycogen, glucose, pyruvate, acetyl-CoA, and then NADH. That sort of.
Excuse the blue, but like that's what it is. Yeah.
Yeah, that sort of makes sense.
Yeah, cool. Thank you.
Yeah, if you guys didn't see the flowchart I had last week, I might refer back to this a couple of times. But yeah, big scary flowchart, big step. Okay, can appreciate that. Okay, so normally at rest, our bodies would use fats, so that's why we store like a lot of fats around our body because it's more energy dense compared to like carbohydrates. So we can basically get fats from one of two ways, right? So we can digest the fats that we eat, right? So we can get like dietary fat from like our food, or we can use our stored fats and basically turn that into energy too, right? So we can either get fat from our food, or we can get fat that we've already stored. And how we get energy from that is, so you might remember me saying last week, if you were there, that like the main purpose of metabolism is to get ATP, right? So you want to get energy, right? So the way we do that from fats is we have to convert it to acetyl-CoA, pretty similar to glucose, right? So we converting glucose to acetyl-CoA as well eventually, and here we are also converting fats to acetyl-CoA, just in a slightly different process, which we'll get to on later. So the exogenous pathway, exo basically means like outside, right? So exogenous basically means the pathway that outside fat, or like dietary fat, the fat that you eat goes through, as opposed to endogenous, which means inside, so like the fat which is like already inside your body. So yeah, so we have fats that we've eaten, right? And when it gets to our gallbladder, the bile in the gallbladder will basically break down that fat into little droplets, right? And why it does this is because if you might remember from chem that like increasing the surface area will increase the rate of reaction, right? So what the bile does is it increases the surface area of the fats that we eat, right? So if you can imagine like that's one big fat molecule, right? And then what the bile does is like breaks it down to little ones. Yeah, so that's going to increase the surface area available for lipase to act on, compared to this really big one, which doesn't have as much surface area. So yeah, lipase would be an enzyme in our pancreas. And that's going to act on the little fat droplets, and that breaks it down to free fatty acids and monoglycerides. Yeah, So, and then after that, our free fatty acids and monoglycerides will basically become combined again to create tags. So that's really important, right? So basically the fats get broken down, but then like they get joined up again, just at the different forms. So they get joined up to triacylglycerides or like triglycerides, right? And what happens after this is that triglycerides, they're packaged up into chylomicrons. What chylomicrons basically are is they're a mixture of like fats and proteins, right? And it's important that the tags are packaged into chylomicrons because they can't travel in the bloodstream by themselves. They're lipophobic. Wait, is that right? Hydrophobic? Anyway, they like
Hydrophobic, yeah.
Hydrophobic, yeah. So like they're scared of water. They can't travel the bloodstream by themselves. So that means they gotta get packaged up with proteins in order to travel in the bloodstream. So that's what the packaging up to chylomicrons does. That's really important, but you just gotta remember that the lymph is involved, I guess. And yeah, they enter the circulatory system, and that helps them to deposit the dietary fat into our tissues. Any questions?
What I really recommend is that because it's a large cycle to try and remember, especially when we get into the next bit of the pathway, it's good to, Ben explains really well like why this happens. So if you understand it, it'll help you remember more.
Yeah. So this is just like this little screenshot of the flow chart, little section. So we can see here that I guess the important part is that the droplets, right? So they get broken down, but then they kind of get recombined as well, right? So I guess that's something you need to remember. Lipase, I guess, I think they quite asked the question like what enzyme is involved and all that. So I guess it's good to remember. So so they get packaged in chylomicrons, and then they can deposit free fatty acids to tissues, which can undergo beta oxidation, which I'll explain later. And then some of the chylomicrons don't get into the bloodstream. They actually go into the liver instead, which I'll go through now. Cool drawing, here. Okay. Yeah, so basically that's what I was going through before. So our body needs to package our fat into little molecules called chylomicrons in order to transport it. So the external core is hydrophilic, right? So that means that like it loves water. So that means that that external core, like the outside, can travel. It will help the overall structure to travel inside the bloodstream. Okay, we'll go through that a bit later. Okay, so remember I was talking about how some of the chylomicrons don't actually get into the bloodstream and deposit fat into tissues. Some of it actually goes into the liver, right? So I actually don't know why it happens, but you just got to remember. Anyway, so in the liver, they get broken down to triglycerides and cholesterol. And what this triglyceride and cholesterol does in the liver is that it forms VLDLs, or very low density lipoproteins, right? So they're sort of similar to chylomicrons in that they're like a combination of lipids and proteins, hence the name lipoprotein, right? So what this VLDL does is that it also serves a pretty similar function to chylomicrons, right? So it's going to deposit some triglycerides and cholesterol into tissues, right? So in the process, it becomes IDL and then LDL. The reason why this happens is because like when you're depositing like triglycerides and cholesterol, right? So a smaller proportion of lipoprotein is going to be lipids, right? So that means that a higher proportion is going to be protein, right? So in other words, there's going to be a higher density of protein, right? So it goes, the density of proteins goes from very low to low. I mean, like it's still like not much, obviously, because it's low, but like it's a higher proportion. If that makes sense. Yeah. Anyway, so
the intermediate density.
yeah. Also, yeah, it's kind of confusing how they put intermediate between very low and low. Like common logic would make you think that the intermediate goes after low. But
yeah, they really like common logic.
Scientists are stupid things. Anyway, so yeah, so this VLDL goes through the process of depositing stuff and then turns into LDL. Now, yeah, that's the last point is, I guess, important to remember. So VLDL is mainly depositing triglycerides, right? And then once they've deposited all the triglycerides, it's LDL. When it becomes LDL, it's mainly deposits cholesterol. So we have questions. Do we have questions in the chat?
Any questions?
Oh no, that's you.
Yeah, that's me. I'm just typing stuff in.
Okay. Right. Okay. Yeah. So that's just a diagram of it. So you can see here that I was talking about the carbon microremnants. So they get repackaged into VLDL, and then they become IDL, and then they become LDL. Now, this stuff here is, I don't think it's very important for semester one. You just need to know that like there's certain like molecules in the bloodstream called macrophages, and they try to like eat the LDLs, right? Because like a lot of LDL is bad. Like you don't want a lot of LDL in your bloodstream because it's just going to deposit a lot of cholesterol. So these macrophages try to eat the LDL, but like sometimes like they can't because there's too much, and then that leads to capillary blockage. But that's not too important to me right now. I guess the important thing is that so the LDL will deposit cholesterol in the tissues, and then some of it will also get reuptaken by the liver. So the LDL goes back to the liver.
Yeah. So Corinne is asking. Sorry, keep going. Just didn't mean to block you up there.
The process can start over again. I didn't mention HDL.
Yeah, we haven't got there yet. But so Corinne is asking, is everything in the slide important, and does it need to be basically memorized to the extent where you can recall it easily unless otherwise stated? Look, the thing is that and the next diagram.
I feel like it's pretty important, yeah. So I guess carbon microremnants get repackaged VLDL, IDL, LDL, and then LDL deposits the tissues, and then and then after that it gets reuptaken. I have like summary slides at the end that kind of might help you to remember, I guess. Yeah, it's basically just like a short summary of everything I'll go through, or I've gone through today. Yeah, I'd say this process is pretty important. So if we go back to this slide, right? So we'll talk a bit about HDL. So HDL stands for high density lipoprotein, right? So there's a high density of protein. And why this is good, right? Is because if we see here, reverse cholesterol transport, right? So this is a high density of lipoproteins, right? So it can take up some lipids or cholesterol from the bloodstream, right? And so basically it decreases the amount of cholesterol in our bloodstream, and then it collects and returns it back to the liver, and the liver can do whatever with it, right? So basically saying that HDL is good for you, and that's why you hear about like good fats and bad fats and all that, right? So the good fats will have a lot of HDL in it, so that's going to decrease our cholesterol. Yeah, because normally you hear the term fat, and a lot of people think it's bad, but you got to remember that it's like there's some certain types of fats that are good, and those fats are referring to like the HDL, which are involved in decreasing cholesterol levels.
Do you have a question, Karina?
And for the like intermediate density lipoprotein, is like they are like kind of like in between, because like HDL is like good and LDL is not good.
So IDL.
It's
So
part of the
are you
packaging
saying?
process.
Are you asking if it's good or bad?
Yeah, and its function.
So its function is pretty similar to VLDL. So it also deposits fatty acids and cholesterol to tissues, basically. It's just another name, I guess. So VLDL and IDL, right? So when VLDL turns into IDL, it's like basically the same molecule, except it has a lower density of fatty acids, right? Because if you can imagine that the VLDL is depositing some like fatty acid tissues, right? So if it's depositing fatty acids to tissues, that means that there's going to be less fatty acid in the molecule, right? So there's a lower density of fatty acid and a higher density of protein. So the density of proteins rises, so its name kind of changes.
The point is that VLDLs, because they're depositing, they have a natural function. By the time they get to LDLs, that's when you don't really want them around, because that's actually what's bad for you. So it's not really a matter of saying whether VLDL or IDL are bad for you. They have an actual function. It's when it gets to that final point and there's a build up and there's too much of the LDLs, that's when it's actually bad for you.
Oh yeah, it's kind of like very generalized, like saying LDL is good, HDL's.
Okay.
I'm sorry, LDL is bad, HDL is good, but like you don't want too much LDL, obviously, because like you're depositable cholesterol. Like some of it's like okay because you want some cholesterol, but generally you don't want too much.
And like the VLDL is kind of like the lowest.
Yeah, that's right. Yeah, so that's the lowest, and then that's kind of like in the middle, hence the name intermediate, and then that's like the worst one of all. And HDL is a good one. So,
Cool.
Nikita put there. Can look at it if you want. I basically gone through most. Okay, yeah, lipid breakdown mobilization. So if you recall me saying at the start, so we can obtain energy from fats in two ways. So we can like the process I just talked about before of exogenous pathway and all that. We can also mobilize the fat that we already store in our body, right? So like the fat around our waist and arms and all that, right? So I don't know what Nikita made this slide. I don't know what that first top one is saying. Anyway, so so first of all, what triggers the breakdown of our fats in our body basically is like so when you're like starting, right? So when you haven't like eaten something in a while, right? So you get higher amounts of adrenaline and glucagon in your body. So those are basically like hormones in our body. And what that does is that it kind of activates lipase, right? So the higher amount of these hormones will activate lipase, and what that's going to do is that's going to break down the fatty acids in our body that we stored to triglycerides and glycerol, right? Actually, so I'll say it again. So a breakdown of triglycerides into fatty acids and glycerol. Yeah, I think that makes sense. So the glycerol is just going to be used in like glycolysis, not too bad. But the fatty acid, right? So what the fatty acid does is that it binds to serum albumin. Serum means like blood, like the liquid part of the blood. Albumin is basically like a protein, right? So it needs to bind. This free fatty acid needs to bind to this albumin or protein, right? Because if you remember me talking about how fatty acids are hydrophobic, right? So they're scared of water, so they can't travel in the bloodstream by themselves. So they need to bind to this protein here, albumin. Similar to like chylomicrons and VLDS, right? So like the fatty acids there in those pathways, like the exogenous and endogenous pathway, they need to bind to the protein to make it a lipoprotein, right? So pretty similar process here. And then yeah, so once that happens, the fatty acid travels in the blood and it undergoes beta oxidation within cells, and that's gonna produce acetyl-CoA, and then it can go through the citric acid cycle and then the vector transport chain. So this is beta oxidation here. I believe you guys had a question on it or something, where you got like 18 carbon molecules or something. Whatever. Yeah. So we'll just go through the quickly again. So yeah, so we have like a fatty acid molecule, right? So like say one of 16 carbons, and then each cycle will reduce it by 2 carbons at a time. And then each cycle will also produce one NADH and one FADH2. So yeah, I think that's much else to say. Okay, so this is the reverse of beta oxidation, right? So beta oxidation, we want to convert fatty acid into acetyl-CoA into fatty acid, right? So, so I guess the important part of this slide, we'll go through this top one first, right? So, you might remember me saying that fatty acid breakdown is increased when there's a lot of glucagon and adrenaline, right? So this is basically the opposite. So fatty acid synthesis is downregulated or like decreased when there's a lot of glucagon and adrenaline. On the other hand, fatty acid synthesis will be increased when there's a lot of insulin or high calorie intake, right? So you can imagine that like if you just had like a big meal, right? Then you've got like a lot of glucose and all that. So you've got a lot of acetyl-CoA. So you don't need like all that energy, right? So what your body says is like, okay, let's take some acetyl-CoA and convert it to fatty acid so that we can store it and we can save some energy for a later date because we don't need all this energy right now.
Yeah.
So
we've got a question about the 2C from acetyl-CoA. So what's that supposed to stand for, Ben? Do we know? Is it important? Is it really important? I don't know.
It just means 2 carbons.
Yeah, 2 carbons at a time.
So
yeah,
2 carbons.
Yeah, it's not that. Yeah.
Yeah. So anyway, the important, apart from the third dot point, the important thing to remember is that before it can turn to a fatty acid, acetyl-CoA has to turn to malonyl-CoA first, right? So this is basically what's called a committed step. So what a committed step means is that once it's converted to malonyl-CA, it can't go back. Right? It's like the point of no return. So I guess that's something you just need to keep in mind. I think they asked us questions in that last year, so like, yeah, so it's good to keep that molecule in mind. The enzyme is really important. This last dot point there is not too important. But yeah, the third dot point here and then this malonyl-CoA dot point here is pretty important. Cholesterol synthesis isn't too important to remember. You just need to know that just like fatty acids, they're produced from acetyl-CoA. So like here you've got acetyl-CoA to fatty acid, and then similar process with cholesterol, so acetyl-CoA to cholesterol. Ketones. We'll talk about that a bit later when we go to. Actually, we talked about it last week. So basically, I'll just go through it a bit again. So basically, ketones are like a last resort sort of energy source. So they're really energy efficient, but they're really toxic to your body. So your body doesn't really want to produce them. But like if we don't have any glucose and stuff, and like if we're starving or something, then our body will produce ketone bodies. It's sort of like a last resort mechanism to give us energy. Yeah. I didn't make this slide, but whatever. Anyway, yeah, good one, Nikita. Anyway, did you just copy this table from PSPeas? This is like for PSP from last year. That was pretty good though. So let me see if there's any important things. Yeah, I guess you just need to remember the heart. Remember like the primary energy source for each sort of organ, right? So the heart and muscle mainly use lipids as a source of energy, whereas the brain mainly uses blood, mainly uses blood glucose. I guess that's the important part to remember. One more important thing to remember is if we look at red blood cells, right? So they use glucose as a source of energy. Now, this is important to remember because in exam questions, they might ask you if red blood cells have like a high or like a low utilization of glucose, right? And a lot of people might think that because red blood cells don't have organelles, and that means they don't have mitochondria. If they don't have mitochondria, that means that they can't undergo aerobic respiration, so they don't use a lot of glucose. That's what a lot of people's logic is. But you need to remember that red blood cells actually use a shit ton of glucose. I think they have like the highest glucose utilization of cells in the body, right? So that's something you need to keep in mind, that even though they don't have mitochondria, they actually use a lot of glucose. And the reason for that is because they don't have a lot of mitochondria, right? So they can't undergo aerobic respiration, but they need to get energy somehow. So they just undergo anaerobic respiration, right? But like that produces like a lot less ATP. So they need to use like a lot of glucose to get that ATP they need. And yeah, because it's kind of counterintuitive if the red blood cells use aerobic respiration, because like they're using up the oxygen that they're meant to carry. So that'd be pretty stupid. So yeah, just remember that red blood cells use up a lot of glucose. Yeah, alcohol. Okay, I know you guys haven't done alcohol metabolism before, so I'll try to explain it a bit more simply. So the main molecule involved in alcohol. Yeah, real funny brain. Okay, so the main molecule in alcohol, which is metabolized, is ethanol, right? So ethanol is type of alcohol. Now, ethanol to acetaldehyde. I guess these parts aren't too important. I guess you could remember the names of acetaldehyde and acetate. They're not too bad to remember, but like that's not really the main clinically relevant part. The main clinically relevant part about this is that in order to get from ethanol to acetaldehyde, right, you need NAD plus, right? Because NAD plus, so you need NADH. You need NAD plus for this step, and then that's going to produce NADH at the end, right? Same with this step, right? So you need NAD plus for this step too, right? And that's the important thing I want you to remember, keep in mind, while you're going through the tube, right? Because NAD plus is a really important molecule for metabolism, right? So, and I'll explain why. I'll just go back to the chart. Okay. So if we look, yeah, I'm not sure if you can see properly, but I guess if you remember, right? So we look at the citric acid cycle, right? So we have acetyl-CoA to NADH and NADH2, right? But in order to produce NADH, you kind of need NAD plus, right? So you need NAD plus to undergo the citric acid cycle. So when you're drinking alcohol, right? So you're like using up a lot of NADH. You're using up a lot of NAD plus to metabolize the alcohol, right? But that's like really bad because if you use up a lot of NAD plus here, you're not going to have enough for here, right? And then if you don't have enough for the citric acid cycle, you're not going to have enough NADH for the electron transport chain. And that means you're not going to have enough energy. And that's really bad. So that's just something I want you to keep in mind, is that alcohol metabolism is bad because it uses up a lot of NAD plus. And I'll go through the specific symptoms and why they all relate to a lack of NAD plus. Right? Yep, so first of all, you get lethargy, or like fatigue. You just feel really tired, you drink a lot of alcohol. So the first reason you feel tired is because you have a lot of lactic acid production, right? And this is because if we look at anaerobic respiration, which is over somewhere. Yep. So what this does is it's going to replenish our NAD plus stores, right? So our body wants to undergo anaerobic respiration to increase the amount of NAD plus we have. But what that does is that it builds up a lot of lactic acid in our bloodstream, and what that's going to do is make our bloodstream really acidic. And what that's going to do is kill off our red blood cells because, like, if it's too acidic, our red blood cells don't really like that. So yeah, that's going to reduce the ability of our blood to carry oxygen and all that to our tissues, and then that means you get tired. Yeah, so that's one reason. The second reason is because, as I said before, you have enough NAD plus to undergo the citric acid cycle, so converting acetyl-CoA to NADH and FADH2, so you get less ATP. And that's the other reason you feel tired. This is another symptom of alcohol consumption, so hypoglycemia. So hyper means low, and then glycemia means glucose, so low blood glucose levels. The reason why that occurs is because so we have pyruvate here, right? And you can recall me saying before that we want to undergo this pathway, so pyruvate to lactic acid to produce NAD+, right? But if all our pyruvate is going through this pathway, there's not enough pyruvate going through this pathway, right? So there's more anaerobic respiration, but there's less gluconeogenesis. So there's not going to be much glucose in our bloodstream, and that might give rise to a whole host of other complications that you might not go through that much detail. Just you just need to remember that like alcohol consumption leads to low blood glucose levels. Weight gain. Yep. So so if you remember that beta oxidation, I think that somewhere. Yeah. So fatty acid synthesis. Yeah. I'll just talk through this. Yeah. So fatty acid synthesis, right? So that uses up NADH, and that gives us NAD+, right? So our body kind of wants to undergo this pathway, so acetyl-CoA to fatty acids, and that's going to replenish NAD+ stores in order for us to do all the other things we want, like the citric acid cycle and all of that. But like obviously, if we have too much fatty acids, then we're going to gain some weight. And beta oxidation is also decreased because we recall that beta oxidation produces NADH, right? And in order to produce NADH, we need NAD+. But if we don't have enough NAD+, then we can't undergo beta oxidation. So there's less fatty acid breaking down, and that means that there's more fatty acid being stored up in the body. Yeah, any questions?
a lot to take in. I think Ben, like on your diagram as well. So on his massive diagram, it also shows like the free fatty acids thing, what it was just showing. So that takes up NADH as well. So if you want, just refer back to that diagram because that has almost everything you need to know.
Great metabolic race. Okay, I did make this slide.
Yeah, Nikita. Nope,
Nikita's
she
looking.
has gone. That's right. We can talk through it. All right.
All right. So I guess it's kind of like a timeline. So if you could imagine you're like a marathon runner or something, then you're having a race, and then this basically goes through basically like the primary energy source throughout each stage of the race. So let's start with before the race, right? So just warming up something. So you're primarily going to be using fatty acids, so because they're really energy dense. So I guess they're more efficient for producing energy. So at the start of the race, your body is gonna start to use a bit of glucose as energy, right? And why that is because like fatty acids are energy dense, yeah, but like they might take a while to break down and provide you energy. You kind of want quick energy because you're like racing and all that. So your body starts utilizing some glucose as well to give you some quick energy. At the start, like from 0 to 2 minutes, it's going to be anaerobic because like your body's still adapting to the exercise. It's not getting enough oxygen yet. And then after a while, like 2 to 5 minutes, your body is able to adapt. So instead of anaerobic respiration, it can start to undergo aerobic respiration instead, and that's going to give you like a lot more energy. So yeah, and then it keeps using up some glycogen and glucose for aerobic respiration. Now at this point, 5 to 30 minutes, yeah, starting to use up some glucose, but like as you can imagine, like the glycogen and glucose might be getting a bit low. And 30 minutes is the important time frame you need to remember, because at 30 minutes, your glycogen and glucose runs low to the point where fatty acids start becoming the primary energy source again, right? This doesn't mean that fatty acids are the only source of energy use. It just means that like the majority of your energy is going to be coming from fatty acids, right? Same with 0 to 30 minutes, right? Like we wrote here is like glycogen and glucose. That doesn't mean that your body's only using glycogen and glucose as energy. It just means that the majority of your energy is going to be coming from glycogen and glucose.
Kind of like how you might hear the thing where people say, "Oh, you use more fat, you use up more fat energy just by being awake or by doing anything else," right? Yeah, it's true, but it doesn't mean that you use up a lot of energy. The fatty acid energy will always be quite similar, and it'll be that baseline. So what's saying is at this point, you've run out of glycogen and glucose to the point where you get the energy amount, let's say, is a baseline, and going from, I guess, your right to left. So from the start here, you have your baseline. Then you get like a graph, and you get glucose and anaerobic pathways usage. That's all this energy you're getting up here. And you start switching to glycogen and glucose, and you still get a high amount of energy. Then after about 30 minutes, you start running out of that energy, and it drops back to the baseline where you start using fatty acids again. So yeah, you can see that in the next graph as well. But that's what's referring to. So the baseline will always be similar.
Oops. So after about 8 hours or so, not sure how accurate protein is.
Oh, so basically, this is the point where if you don't have any fat, and this applies especially to skinny people who don't store fat well, or like if you have a condition, right? This is where you might hear something like your muscles start to break down to provide energy for your body. That's because you have no other energy left to give. And that's the point at which you are starving, or starvation, and it's quite a serious issue. So that's what it's referring to, basically. And what happens is it goes through the pathway where you want to build glucose or glycogen from whatever you can get your hands on. And that happens to be, there's no fat, so usually that's what you go for first. No fat, then you get to protein. You start breaking that protein into into amino acids, and then that breaks it back down into pyruvate, and then back into I think glucose, and then starts using that for acetyl-CoA again.
Mm-hmm. Yeah, that's right. Thanks for helping me out. Yeah, so you basically like run out of fatty acids and glycogen to use effectively. So your body kind of is just like, yeah, we're going to break down some proteins in muscles to give us energy. Okay, this final point is really important. They've asked us questions on it in the past. So whenever you see a question where it's like this guy sprinting, all right, or like yeah, like sprinting or something like that, or like another like short burst of energy required. The main molecule used in that is creatine phosphate. Yeah, so I guess that's something you need to remember. I don't really know why.
What's actually interesting, I heard from another first year of yours, a friend of ours, Aditya, actually said like he apparently in the AL he asked about creatine phosphate, and some of the AL tutors said no, it's not really important. But for us, it was definitely common knowledge that you use creatine phosphate for things like the fast, high energy sprints when you need to use that, and that's what your body quickly reaches for. If it's not going to be glucose and you need to use up a lot of energy for a very short amount of time, it's going to use up creatine phosphate. So that's something that we believe you should remember as well.
Yeah, thanks, Brent. Okay, so this graph might seem a bit confusing. But basically what this graph is trying to show, so we're going to ignore the parts before breakfast. Right, so let's say you've had some breakfast, right? So after you had some breakfast, your main energy source right after is going to be dietary carb intake, right? Because like, as you can imagine, you just had a good breakfast, so there'll be carbs being digested and all that. Then a few hours pass, and after that, your main source of energy is going to be glycogenolysis. So basically means you switch from having the glucose that you just ate as your main primary energy source to the glycogen that you've stored in your body beforehand as your main energy source, right? So yeah, so from dietary glucose to glycogen. Then after that, so you've run out of glucose and you run out of glycogen. That's when you start to rely on gluconeogenesis, so the thing that we were talking about before. So you might rely on proteins and all that. So obviously you don't really want to get to this stage because like you might lose some muscle and all that. Yeah, so that's basically.
Usually fats will be used before proteins, right?
Oh yeah. I don't think it's on this thing. Yeah, but proteins are kind of like a last resort sort of thing. Oh, second last resort to ketone bodies. So yeah, dietary glucose to glycogen to fats and protein.
Yeah, that's why if you get older, you might start fasting because your metabolism might not be as quick. So it takes longer for you to use up the glucogen and glycogen in your system before you move on to fats. So if you eat less frequently, you tap into the fat resources quicker and burn that fat.
Yeah. So this is just a bit about what I was talking before. So we can see this graph, right? So before the start of a race, right? So x-axis should say like minutes, like after like the start of a race. Yeah. So anyway, so we can see that like before the start of the race, the fats will be somewhere around here and carbs will be somewhere around here, right? So that's basically saying that fats are the predominant source of energy before a race. Then once that race starts, you can see that carbs goes way up. So your body starts using carbs and glycogen as a source of energy, right?
And remember, it's percentage, not amount.
Yeah. So I feel like I think like the total amount of fat used is actually gonna increase too because you want energy, right?
Yeah, you do want energy. Yeah.
Yeah. So it's just that the proportion of fat in relation to carbs decreases. So both carbs and fats energy like usage increases, just that like the proportion decrease for fats. Anyway, so yeah, and then once so the race kind of progresses and then you start running out of carbs, then you reach this point here, right? So the magical 30-minute barrier. And that's when you're going to start using fats as the dominant source of energy again, whereas your carbs have run out. So like the proportion of carbs that you use is going to get lower.
Yeah.
That's basically what that diagram's saying. And yeah, we're finished. These are summary slides you can go through by yourselves. It's basically summarizing the slides that I've gone through today.
Yeah. Does anyone have any questions about what we've just gone through? And if you guys are watching and you're not watching live, people who are watching, feel free to drop us a message, Ben or Nikita or myself, and we can gladly help you out. But yeah.
And Ben has written a lot of practice questions about this topic, right? So that will be in our week 4 revision session. So if you guys want some consolidation on this, feel free to drop in on that session.
Yeah, that might be a collab too with another PSP.
Yeah, so we might
quick nudge. Anyway, I guess the difference between summary and high yield exam points is that these are kind of like the main concepts that are important to remember. These are like really they're not as clinically useful, but they're sort of just like random facts that you sort of need to like.
They're going to test you on,
yeah, for exams. So these ones are more, I guess, clinically relevant, more conceptual based, but these are ones that are just random facts. But both the summary and the high yield exam points would be good to remember. So yeah, if you took away literally nothing from these slides beforehand, just look at these last 4 slides, I guess, and there should be a good summary.
Do you need to remember the drugs in the active learning session? So which learning session were you talking about?
Also, what drugs?
What sort of drugs?
Yeah, what drugs? Yeah, what drugs were
the
these?
One about blood glucose, like homeostasis, essentially.
Oh, central dogma.
How the fuck is it 241?
Okay. Is this about how the bacteria and viruses enter the system and you use antibacterials against the bacteria?
Carbon monoxide.
Oh shit! I'm still streaming. Oh, my bad.
Carbon monoxide is important because it's to do with how carbon monoxide binds to hemoglobin more than oxygen does. So if you have carbon monoxide poisoning, that's basically like you're being asphyxiated. Basically, it's being like your body's being deprived of oxygen because hemoglobin can't bind onto oxygen. So that's how I know about carbon monoxide. So yeah, I would say that's important. I don't know what rotenone is though, but that might be because I don't remember stuff.
Might be good to kind of Google that.
Yeah, quick Google.
Rotenone.
Yeah. Thanks for sticking around, guys. I didn't realize I went for that long. What the hell? It's 242.
That's all right. Yeah.
Thank you.
This stuff does take a while to like get the information out. So
I know Biochem's really boring.
It is.
It's
sometimes, but it is quite.
You guys are first years.
It's worth it.
Rotenone is not something that I know, so I would say don't worry too much.
Rotenone.
Yeah. Anyway, Biochem's.
We had to fill out an entire table, and we just had to write about which enzyme was inhibited by each drug and the impact that would have on like different metabolic pathways.
Definitely not. Okay. You definitely.
Yeah.
You don't need
to. No, you won't need to know that.
Yeah. That's not something we learned last year. That wasn't on the actual learning exam objectives. And even if that is for your AL, I don't think it will be something that's testable in the exam because it's far too in detail, and drugs that they're not drugs on the actual list that you need to remember. We have like a list of how many drugs. They're not on those. They're not going to do that.
When it comes to drugs, I would focus on the pharmacology lectures which you're going to have. At the start of each lecture, they'll give you an e-pharmacology drug list, and usually those are the drugs that you can be tested on the exam. These ones are just conceptual. I think what they want you to understand in that learning was that if you're inhibiting something, it's going to stop another pathway from happening. So try and just think of it like that more than memorizing.
So rotenone interferes with the electron transport chain. And I hope I'm not breaking out here.
Out of curiosity, was that AL activity about the enzymes in the mitochondria?
I think it was the role of liver in metabolism. That's the AL I have up now from last year, but I'm not entirely sure.
All right.
Okay.
Cool.
Yeah.
We're still rolling.
But I mean, I don't remember having to answer a question about rotenone on the exam, so I don't think that's something you need to remember too specifically.
Yeah.
Do you guys have any tips on learning all the pathways? Good question.
mind map. So what I did was I grabbed a whiteboard and I grabbed a friend, and basically what we did is from another PSP which taught us, I kind of broke everything down, and in the slides you would have seen my drawings, really bad, but they kind of display a pathway. So if you can have a go at drawing it out a few times, I can upload that to the PSP resource folder if you want to. I have metabolism. I've done the whole unit on a whiteboard and basically summarized it into drawings or words. So that's what I did, and that worked for me. Other kids do other things.
Yeah.
The best way, is to if you can explain it to someone else, then that would be the best. If you want to help someone else, get them to explain it to you. Like that's the fastest way to learn these conceptual pathways when there's so much to actually remember. It's the best way to learn it and understand it.
And that's why we're doing revision sessions as well. So you guys get a chance to do that.
Yeah. I like to learn visually, so like I made all those flowcharts, spent like a couple of hours on the flowchart, metabolism. So that's basically how I learned. Feel free to kind of use that as a starting
Ben
point.
used to put that on his desktop and just like look at it as he was doing his work. And it was like
it just goes through osmosis.
Exposure helps.
Yeah, osmosis, just osmosis into my brain. Thanks for sticking around, guys. Like honestly.
Yeah.
High concentration, low concentration.
Anyway, all this biochem stuff, like, who really gives a shit about biochem? All we care about is HKS. Go read up HKS notes.
That's true.
Share them around with your friends, because
no one wants to read HKS.
But
nobody does
that we got.
Except me.
Well, we don't want you guys to suffer through the same pains we did, so please spread them around. Hopefully, Jaffies can learn from our mistakes.
All right, spread the word.
Sorry about the background noise.
That's all right. I think it should be fine. But yeah, so hopefully next week we'll be able to run on Facebook as well or YouTube live, so more people will find it more accessible. And we'll catch you next Wednesday, I hope, for another content session. We'll be moving on to the starts of your cell anatomy and a lot of other things such as, let's have a look, biotech, epithelium. We'll be starting epithelium, and that will be pretty fun. So catch you next time.
Keep your eyes out for revision session 2 on Friday. So next week's revision session as well. So Wednesdays and Fridays hopefully, and if not, we'll schedule another time. All right. HK's pretty ready to play.